Okay, wow, that was quick. So, for all people out in TV land, I just passed out the note cards, and if you didn't get one here today, you'll need to come see me in my office to get a note card, and that's fine, but you will have to get one from me. As I said before, everybody will need to bring a note card to the final exam that you get from me, and if you bring a note card that's not from me or you don't bring a note card, you're going to lose points. So please make sure that you have your note cards and you bring it to the final exam. For those of you coming in late, I will have some available after class if you want to come grab me real quick, and I can get you some note cards then. Okay. Today I'm planning to get most of the way through the last of nitrogen metabolism. As I mentioned to the class last week, Friday's lecture will be my last lecture to a big class. So I've got some surprises planned. And I think it's going to be fun, so I hope that you all will come. I probably, depending on how much time I have, will wrap the review session into that Friday session. So if you're wanting a review session, Friday is probably, depends on how far I get today, is probably when I'm going to do a lot of that, and we'll see that on Friday. If not, then I'll schedule a separate review session. And of course, as always, I'm happy to meet with you. My schedule is kind of chaotic right now, so if you want to meet with me and you find that you can't find me, just send me an email and I will schedule an appointment with you to meet with you. Before I get to that point, I'll say how much I really have enjoyed this class. You guys have been a wonderful group to work with. I've gotten to know many of you from the class, and it's been a lot of fun. So I've really enjoyed that, and I hope that you have as well. All right. Well, that said, let's dive into a bit of nitrogen metabolism. So last time I showed you how nitric oxide could be made, and in fact is made inside of cells as a molecule for signaling. And I noted that one of the signaling processes it's involved in is vasodilation. Nitric oxide is a fairly reactive substance. It's one of what we call the reactive nitrogen species. And yes, I've changed the order of these things a little bit. I see people flicking through their figures. I changed the order just a little bit. I did that to simplify things because I had a little bit more material in there than I really wanted to talk about. So I got rid of a couple of figures that you had in the original notes. Anyway, so nitric oxide is a reactive oxygen species, and we know that when we have reactive molecules inside of cells, that's problematic for cells in many cases because they tend to glom onto other things in a non-enzymatic fashion, and non-enzymatic reactions for cells are almost always problematic. But what I'm showing you on the screen here is one of the very problematic reactions associated with nitric oxide. And it might seem odd that you've got a molecule in the cell that is reactive, but that's what cells have evolved to do. They don't make a lot of it, but they do make some. And it's important, as I said, for some signaling processes. Nitric oxide can react with another reactive oxygen species called superoxide that I've mentioned before. And superoxide, when it combines with nitric oxide, makes a very nasty compound called peroxynitrite. Now I'm going to show you a fair number of molecules and reactions today, and I'm not going to ask you to reproduce these reactions in any way. The names of the molecules are sufficient, okay? The names of the molecules. So yes, you should know what nitric oxide is. Yes, you should know what superoxide is. And yes, you should know you put them together to make peroxynitrite. Peroxynitrite is a molecule that can react in a whole bunch of ways, and I'm not even going to bother to talk about all these other ways that are here. But I will tell you that peroxynitrite, because it is so reactive, can commonly react with proteins and cause problems. And we'll see this with some of the nitrites in general, that they are very reactive molecules that can react with proteins. Some of the things that can happen when these molecules react with proteins include nitrosylation, that is putting a nitrate or nitrite onto an amino acid. And so one of these amino acids that commonly gets a nitrate put onto the end of it is tyrosine. And if you think about it, that would drastically change the charge of that tyrosine at physiological pH. has an R group that is uncharged, but you put a nitrate on there at physiological pH and it's going to be negatively charged. So when you think about how that might change the structure function of a protein that it attaches to, you can imagine that the consequences could be fairly significant. And that's indeed what happens. Another amino acid that can be affected by these processes that I'm going to describe to you is cysteine. And cysteine can be readily oxidized, not even necessarily reacting with nitrogen, but instead oxidized and cause some problems as a result of that. Nitrites can form in the body, and nitrites are molecules that form by the ionization of nitrous acid or by the reduction of nitrates. So one of the concerns about fertilizers and nitrogen fertilizers and things that go on to various food substances is the presence of nitrates. I used to live in the country for a period of time, and I know that when I moved there, one of the first things they did was to approve the sale of the house, they had to test the water for nitrates, because nitrates in the water can get there from nitrogen fertilizing and so forth, and nitrates in the water are problematic for some of the reasons I will describe here. And you can see that it's not uncommon to get nitrates and nitrites actually in your diet. So I don't want to leave everybody the impression that, oh my God, I'm going to die if I eat a vegetable that's got something like this on it. But the point is you want to reduce your intake of compounds like this. Nitrites are commonly used to cure meats and to prevent botulism. So if you eat bacon, for example, you get a pretty fair dose of nitrites if it has been treated, and probably about 95% of the bacon has in fact been treated that way. It's rare to find untreated bacon around, and of course you have to be careful in the handling of any of these compounds if they haven't been treated. Nitrates can be reduced to nitric oxide in hypoxic conditions. What do hypoxic conditions mean? It means low oxygen concentrations. So your body goes through hypoxic conditions anytime that the oxygen use is greater than the blood supply can deliver. So hypoxia is not an uncommon phenomenon. So the more nitrites you have in your body, the more likely you are to form nitric acid, and that, nitric oxide, and that could pose some severe problems. 90% of the nitrites that you get actually come from your diet, from the reduction of nitrates in vegetables. And these, as I said, come from fertilizers or plant stress. Stress plants can also produce nitrates. And let's see, the last thing I want to point out about this is that nitrites readily form nitrosamines, and I'm going to show you that in just a second. Some of the consequences of the oxidation that come from nitrites are that you can oxidize things that you don't want to. So for example, the iron in hemoglobin. If you oxidize the iron in hemoglobin, you convert it from an iron +2 to an iron +3. An iron +3 cannot carry oxygen. That hemoglobin has been rendered useless if it interacts with nitrites. Okay, now, so what am I doing? Nitrosamines are things you really don't want to mess with, okay? So here's a nitrite, and again, I'm not worried about the structures here. Nitrite in the presence of acid, like stomach acid, will produce this nitrous acid ion, which can spontaneously come apart and form a nitrosonium. That nitrosonium ion can react with, as you see here, secondary amines and form a nitrosamine. So where do you see secondary amines? How about in peptide bonds? Okay, so you could imagine that we're going to have some real problems that might arise from the presence of these compounds. They would be very reactive, and they can react with amino acids. They can also react with nucleotides, and so some severe consequences arising as a result of nitrosamines. The formation of these compounds is found in things like processed meats. We are increasingly finding that processed meats are associated with some detrimental health effects. These include things like bacon, I've already mentioned, ham, beef jerky, things like that that are very processed meats, can be problematic and be rich sources of the precursors of these nitrosamines. Beer, unfortunately, is also a source of this. Cigarette smoke, chewing tobacco, any tobacco products can form that, and I'll show you a figure for tobacco stuff in just a second. The good news is that the vitamin C, which is an antioxidant, does have somewhat of a protective effect against some of these reactive nitrogen compounds. Question? Yeah, where? Does that mean that nitro beer will have more nitrosamine? That's a very good question and a very good concern, and I can put that fear to rest for the most part. Nitro beer is using nitrogen, molecular nitrogen, instead of carbonation. And so that molecular nitrogen is not very reactive, and so as far as I know, no, there's no effect with that. Do you like nitro beer? I hate it. I hate nitro beer. I really do. I like more bite in my beer. So, but no, you shouldn't have any concern with that. Yeah, but you still get nitrites from beer. Okay. You also get a lot of sulfites from beer, more so from wine. So sulfites are problems commonly for asthmatics. It's almost impossible to find wine without sulfites because it's needed to preserve the wine. Sulfites in high enough concentration, they're also used a lot in salad bars for protection. Sulfites, you're not responsible for this, I'm just rambling as I'm talking here. Sulfites are also able to trigger asthmatic responses in some people with asthma, and I have asthma as well, so that's why I know about this. Okay, now, the biggest concern for nitrosamines, okay, is that they can form what are called DNA adducts. We saw earlier when I talked about DNA repair that we can have these big honking molecules that can link to a base, and I didn't tell you how that can happen. Well, this is how they can happen. You have reactive nitrogen species that can combine with other large molecules and ultimately glom onto a base and form a covalent bond. That covalent bond is problematic because if the DNA polymerase tries to replicate, it can't copy because it can't read that base. And so a couple things can happen, one of which is mutation, which is obviously not desirable. And it's why you want to avoid things like diesel smoke, which is also full of this stuff, cigarette smoke, which is full of this stuff, or any more of nitrites than you absolutely need to have to eat. I love bacon, by the way, so I'm not talking down bacon. Let's see, what else do I want to say? Evidence for gastric and esophageal cancer risk as a result of nitrosamines and the products that happen from those. NNK is and there's also NKK is a molecule that is derived from nicotine, that is a reactive nitrogen species, and you can see that there. You're not responsible for the structures of these, but I think that the names of everything that I've mentioned are worth knowing. Okay, well, we could talk about reactive nitrogen species all day and not cover the subject, so I just wanted to say a few things about them, focusing mainly on the nitric oxide, the nitrites, and the nitrosamines. And so I think that gives you a general overview for all of those. The last topic I want to talk about in the course concerns amino acid metabolism. And amino acid metabolism, the reason I leave it to the very end, is very different from all the other metabolisms we've talked about because it's not one pathway. We talk about sugars, they ultimately converge on glycolysis and the citric acid cycle. If we talk about fatty acid oxidation, we have beta oxidation for all the fatty acids that then go into the citric acid cycle as well. If we talk about I guess fatty acids and carbohydrates are the two biggies. If we talk about proteins, the breakdown products of those proteins are amino acids, and they don't have one metabolic pathway. They don't have one metabolic pathway either for their synthesis or for their breakdown, although we will see there is some convergence in the breakdown pathways, although I've simplified that a lot in getting to show you the convergence. First, I want to focus on the synthesis of amino acids. There are 20 common amino acids, and I'm going to add one more into that mix, a 21st amino acid that I'll mention along the way. And they are made in what we call families of pathways. Families of pathways. You see a list of 6 different pathways here. There's really only 5. The sixth one, which says histidine, histidine is in a pathway all by itself. So histidine actually has the most complicated pathway of any of the amino acids. But all the other amino acids can be grouped into the various groups that I'm going to show you here. Now, you are not going to be responsible for detail in these pathways as they relate to this goes to this, goes to this, goes to this, okay? That's not my purpose in showing you these. I do think you should know the groups, and I do think you should know some of the members of those groups, and I'll talk about those as we go along. If I don't talk about an amino acid, then you have no responsibility for that, okay? So let's go through and talk about then the groups, the families of amino acids, and understand those. Previously, I've mentioned essential versus non-essential, and I think you know those definitions as such. And I've also mentioned that essential versus non-essential varies in humans, and I gave an example last time, actually, of arginine, which was in that category. Okay, well, let's talk about the glutamate or alpha-ketoglutarate family. You can use those terms interchangeably because you can see that they all derive ultimately from alpha-ketoglutarate. So whether you call them the glutamate family or the alpha-ketoglutarate family, I don't care. But you should know that the starting material for this whole family is alpha-ketoglutarate. And alpha-ketoglutarate, I hope you remember, is an intermediate in the citric acid cycle. Well, alpha-ketoglutarate, I've mentioned before, and I'll remind you here, can be transaminated. And when you transaminate alpha-ketoglutarate, you get the amino acid glutamate. And glutamate is the precursor of all these other amino acids that are there. The only one that I'm really caring that you know is related to glutamate is right beneath it, and that's glutamine, which again we've already talked about. You substitute the oxygen on the carboxyl group of the R group of glutamate, and you create glutamine. And we talked about that with respect to the movement of nitrogen in the body. So this is a very simple pathway in this sense. It is a transamination pathway. It is important for the movement of nitrogen in the body. You can see that transamination here. This was a figure I showed last time that I'll remind you of what's happening in transamination. There was alpha-ketoglutarate. In this case, the transaminating molecule was aspartic acid, and we can see that the amine of aspartate goes on to the alpha-ketoglutarate to make glutamate, and the oxygen of alpha-ketoglutarate goes on to the aspartate to make oxaloacetate. So we've just basically swapped the amine and the double-bonded oxygen. And that's what happens in a transamination reaction. Those reactions, of course, catalyzed by enzymes known as transaminases. Glutamine synthesis, you may recall in a more complicated figure from last time that basically showed you that glutamate goes to glutamine, and this enzyme I mentioned last time that catalyzes the reaction is known as glutamine synthetase, and yes, that's an important enzyme. That enzyme I haven't shown you, and you'll thank me for not doing that. That enzyme, glutamine synthetase, is regulated in one of the most complicated regulation schemes of any enzyme that I'm aware of. There are probably a dozen compounds that allosterically affect that enzyme. And they're very important because, again, for the purposes I've described to you, the balance of nitrogen in the body and in the cell is very important. And so the synthesis or the breakdown of glutamate and glutamine is very critical in that process. So controlling this enzyme is very, very important. The other important thing about this enzyme that I'd mentioned last time and that you should know is that it is a source of scavenging ammonium ions. So remember that from last time, that we're picking up those free ammonium ions, ammonia molecules in water. And those, of course, are toxic. When they get put onto glutamate to make glutamine, you're essentially detoxifying the water that contains them. So that's an important consideration. Arginine pathways, I'm not going to mention that. You're not responsible for that. The serine family is shown in the upper right. And serine is an important intermediate. It comes from this molecule 3-phosphoglycerate. And that may sound slightly familiar to you. I'll tell you where you've heard it before, and that was in glycolysis last term. 3-phosphoglycerate is an intermediate in the glycolysis process. And in this case, all three of the molecules, or all three of the amino acids derived from serine, are worth knowing. And I'm going to say something about each one. So serine, of course, is one of the amino acids. It has a hydroxyl side chain. You can see its synthesis there, and again, I'm not holding you responsible for it, but you can see here's 3-phosphoglycerate here, and down here we've got serine with its hydroxyl group on it. I'm a little more interested actually not in the synthesis of serine, but what happens to serine to make other molecules. So one of those other molecules made from serine in a very complicated set of reactions is cysteine. We can see serine actually entering here. And you also note that, if you remember, cysteine has a sulfur. It has a sulfhydryl group. So where does that sulfhydryl group come from? The sulfhydryl group for cysteine ultimately comes from methionine. It's a complicated set of reactions. Again, you're not responsible for those reactions, but that's the two amino acids that go into making cysteine are methionine, which is the source of the sulfur, and serine, which is the essential backbone of the molecule. You also notice here that in the synthesis pathway that ammonium ion is released. Okay? So I told you again, this is a toxic compound, and so we are concerned not only where it's used, which detoxifies, but also where it's released, and here's one of those places where it is released. Blah, blah, blah. Okay. Actually, did I not have glycine? Where's my glycine? Oh, here it is. Okay, I skipped this one. I shouldn't have skipped this one. All right, now this one looks very mouthful complicated, but it's really not. Serine has a CH2OH that's not found in glycine. If you were to chop off that thing right there, you would end up with glycine. And that's what happens in the synthesis of glycine. This CH2OH gets chopped off, and it gets donated to a folate. Remember, folates are things that carry one carbon for reactions. You saw folates, for example, in the synthesis of thymidine. So one of the ways you get that one carbon molecule onto a folate is by this reaction in the synthesis of glycine. So from serine to glycine, one carbon goes onto a folate. Don't worry about the names, but it goes onto a folate to make a folate that has that extra carbon, and that carbon can be used to donate to other molecules. This reaction, you'll notice, is reversible. So if a cell has plenty of glycine and it has plenty of folate that has that one carbon unit, it can make serine. So glycine and serine's synthesis is intertwined. Whichever the cell has more of, it will make the other of. Okay? And the cells are quite capable of doing that. The last amino acid I want to mention here is an odd one. And it's one I don't think I've mentioned in the class so far, so I'll take a minute and describe it. This is an amino acid you may not have heard of. It's called selenocysteine. And selenocysteine is commonly described as the 21st amino acid. We know that there are 20 amino acids that make up proteins, so what is the 21st amino acid? Well, it turns out the 21st amino acid makes it into some proteins as well. But there's only codons that code for 20 amino acids. So how does selenocysteine get into proteins? And it's kind of a complicated, I keep using that word today, kind of a complicated system. But suffice it to say that cells have an odd transfer RNA that can carry selenocysteine. By the way, the term selenocysteine comes from the fact that it contains an atom of selenium. There's an atom of selenium. So if you look at, for example, cysteine, and you take out the sulfur and you replace the sulfur with a selenium, you have selenocysteine. Now, there are some odd amino acids that float around the cell that can actually pair with a stop codon. Sort of seems to defeat the purpose of a stop codon. I see some puzzled looks. They can actually pair with a stop codon. And under rare circumstances, a stop codon is not read, a particular stop codon is not read as a stop codon, but instead incorporates a selenocysteine at that point. Now this seems very odd, and it is very odd, okay? Because it goes in with low efficiency, and there are some important proteins that contain selenocysteine. Okay? Well, why do cells have selenocysteine? It turns out the chemistry of the selenium atom is unusual and useful for the function of these proteins. So one of the proteins that and you're not responsible for this, I'll tell you what it is. It's called thyroidoxine reductase, which is an important enzyme, and that enzyme contains selenocysteine. Okay? So it's not, I mean, it's a rare, it's unusual, but it gets in, and it gets in consistently at the same place. If it doesn't make it in, then that protein is not going to function properly, and in fact it gets destroyed if that happens. So selenocysteine comes from serine. All right? Don't worry about the reactions, but know that selenocysteine is in the serine family. Okay, we're moving through these. The aspartate family, you can see it listed up here. The aspartate family all is made from ultimately oxaloacetate, and I've mentioned this a few times during the term, so you should, I hope, remember that if you transaminate oxaloacetate, you create aspartic acid. Aspartic acid being one of the 4 amino acids, and you can see 4 different amino acids that are made from aspartic acid. Okay? The only one that we're going to focus on here is actually asparagine, because asparagine can be made by another important transamination reaction that I'm going to show you. Transamination is not at all unusual here. You can see, for example, oxaloacetate transaminated with an amine from glutamic acid yields alpha-ketoglutarate because it's lost its amine, and aspartic acid, which has gained the amine. That's the reactions I've shown you previously going in the other direction. That's one way of making aspartic acid. I can also make aspartic acid by breaking down asparagine. So you see this aspartic acid going to asparagine here. That reaction goes the other direction. It's not really reversible. I'll show you the considerations for that in a second, even though the arrow shows it's reversible. But the main point I want to show you is if you go from asparagine to aspartic acid, then look what you release. You release, again, toxic ammonium ions. So those have to be mopped up in some way. So let's take a look then a little bit more closely at the interactions between the aspartic acid and the asparagine in their metabolism. So really, in the metabolism of these 2 amino acids, we have to consider that the synthesis really goes one way and the breakdown goes the other way. And they're different things. Synthesis by transamination, as I've shown you. Okay? And this is a transamination, again, a source of amine coming from glutamate. But in this case, instead of making aspartate, we're putting the amine onto aspartate to make asparagine. So asparagine is the equivalent of glutamine. Remember, glutamine had that extra amine group on it on the R group. Asparagine has an extra amine group on its R group. And the donor for that is glutamate. So asparagine synthetase, you don't need to know that name, but asparagine synthetase catalyzes that reaction, and it is a transamination reaction. Notice that this reaction is very energetic. It requires a lot of energy. And you can tell that by virtue of the fact that the product is AMP, not ADP. That means an AMP, whenever you make an AMP from an ATP, that it's the equivalent of 2 ATPs to make that. Okay? So a lot of energy goes into making this asparagine, which means that this reaction is essentially irreversible. This reaction is not readily reversed. So when you've got asparagine, you've either got to use it to make proteins, or you've got to break it down. And when you break it down, you make ammonium ion. Okay? So these are considerations. Cells are careful not to make too much asparagine, because if it makes too much, all it's got to do is break it down. And what it's done is it's converted an amine from here into an ammonium over here if it goes through this whole process. So that's not a good thing for cells to do, so they're careful in how much asparagine that a cell will make. All right, we're moving along. Now we get to kind of a fun family. Not fun in terms of synthesis. The synthesis pathway for the aromatic amino acids are, to be quite honestly, a pain in the you-know-what. So we're not going to focus on that synthesis. We're going to focus more on what the amino acids are actually used to make, because that's considerably of more interest. Tryptophan is a precursor of the various compounds that you can see here. Okay, let me think about that. Yeah, I'm not so concerned that you know this as a list. Auxins are one. They're plant growth hormones that I think are worth knowing coming from tryptophan. The others I'm not quite as concerned about. But the more interesting ones are tyrosine and the catecholamines, which I'll talk about in just a minute. Let's look at the scheme, and this is a very simplified scheme by how these guys are actually made. The important thing off of this scheme is shown right here, the precursor. So the precursor for the synthesis of the aromatic amino acids is phosphoenol pyruvate. And there's a lot of reactions that require getting down to the very bottom that you can see here. You'll notice that I'll show you, I think in a slide after this one, that this shows the independent synthesis of tyrosine from precursors. Tyrosine can also be made from phenylalanine. So this molecule here, you point an arrow to the left, you'll also see that tyrosine can be made from phenylalanine. Tryptophan has its own synthesis, as you can see. All right. What do I want to say? Phenylalanine and tyrosine pathways overlap, which is what I said. And hormones and neurotransmitters, which I'll show you in just a second, are made from these compounds. Okay, this shows you some of the molecules that come from tryptophan. I should have put the structure of tryptophan up here that you can see it's related, but this melatonin is fairly closely related to the structure of tryptophan. I'm going to just talk you through that, and as I told you, the only one I'm going to hold you responsible for knowing are the auxins. Okay? Melatonin is a molecule involved in circadian rhythm, so you can just listen if you want to relax at this point. It affects mood, sleep, and blood pressure, and its production is affected by blue light. So this is a compound that people worry about in terms of getting enough made so that you get sleepy at night. So if you're like me, I have this little screen thing on my computer that turns my essentially blue light off after about 8 o'clock at night so that I'm not blasting myself with blue light and allowing melatonin to be synthesized so that I can get to sleep. And I think it actually has some kind of a positive effect. Has anybody else noticed that? Do you notice an effect? Yeah, I mean, it did seem to have an effect when I activated it, and if you haven't activated that on your computer, I encourage you to do so. I know Macs will do it, and I'm pretty sure Windows will do it too, try activating that and see, especially if you have sleep issues or not, to try that. And of course, if you want to stay up all night for an all-nighter, maybe the thing to do is turn it on, right? But it might make you grouchy or something too, so you've got to be careful about that. Serotonin is a neurotransmitter, causes vasoconstriction, has effects on memory, learning, and contribution to happy feelings. So tryptophan, the happy drug, the sleep drug. Niacin, of course, is vitamin B3. Don't want to say too much about that. Auxins come from tryptophan. As I said, I'll hold you responsible for that. They stimulate cell division and rooting in plants, and they have some interesting effect on plant growth. And there are a variety of these auxins. I'm showing one of them on the right. You don't need to know the name of that. Just know the auxins as a class of molecules. Okay, phenylalanine is interesting mostly because of problems arising with its breakdown. So phenylalanine, of course, is an aromatic amino acid like the other two in this category. Phenylalanine, you can see, can be made into tyrosine, and in fact, the conversion of phenylalanine into tyrosine is essential for the breakdown of phenylalanine. People who lack enzymes that allow them to break down phenylalanine have some severe problems with the artificial sweetener known as NutraSweet. And in fact, they have severe problems anytime they have too much phenylalanine in their diet. NutraSweet is a dipeptide made from phenylalanine and aspartic acid. And it's used in high enough quantities in artificial sweetener that it can be essentially toxic or very damaging to people who have the disease known as phenylketonuria. So phenylketonuria arises because in people who have an inability or lack the enzyme to break down phenylalanine. And so they must restrict their diet very carefully. They can have some severe nerve damage that can happen, severe brain damage that can happen if they get too much phenylalanine. Fortunately, if they monitor their diet and they're very careful with what they eat, don't have to, they can live a normal life, but have to be very careful about the intake of phenylalanine. Now, the most interesting of the aromatics, I think, is actually tyrosine. Tyrosine is a precursor of several molecules known as catecholamines. That's a name I think you should know. Catecholamines are molecules that have a couple of hydroxyls on that benzene ring that came from tyrosine. So if we see here, for example, here's tyrosine on the upper left. It has one hydroxyl group on its benzene ring. All these other molecules have two hydroxyls on their benzene ring, okay? And that's the group of molecules known as catecholamines. These molecules are neurotransmitters, and they're very important for various functions in the body. Dopamine is a compound that is involved in mood, a lot of things associated with dopamine, including Parkinson's disease. And Parkinson's disease arises as a result of problems with dopamine, the synthesis of dopamine. And so people who have Parkinson's disease are very commonly treated with this compound called L-DOPA, because L-DOPA is a precursor of dopamine and can, in high enough quantities, be made, at least in limited, the production of dopamine can be increased in people by dosing them with L-DOPA. Epinephrine you've seen before. Epinephrine is the hormone, of course, that's also known as adrenaline, and it comes from metabolism of tyrosine as well. A related compound known as norepinephrine is very much like it, and its effects are also very much like epinephrine. Okay, so a lot of stuff there. What do I want to say? That just sort of summarizes some of the things I've told you there. I'm not going to go through and summarize that again. The next group of molecules related to tyrosine metabolism are actually thyroid hormones. Okay? So thyroid hormones are a class of molecules that contain iodines on their side chain rings that you can see, on their aromatic side chain rings that you can see here. Now, this is a pretty complicated system, and I'm not going to take you through all that, other than to remind you of the structure of thyroid hormones as the essentially only molecules in the body that contains iodine. And that production, or the starting material of the tyrosine for their production is very, very critical. Yeah, I think I'll just leave that at that. Questions? I'm just rambling. I'm going through fairly quickly. Everybody looks tired. Everybody's tired by this point in the term. A lot of this is just information. I'm just kind of dumping from my head to your head. I hope it's going into your heads. I hear some laughter at that. Oh, there we go. There's some more selenium-containing enzymes. All of the known deiodinases, which are compounds that remove iodine from a molecule like this, are known as deiodinases, and they all contain selenium, gotten there by the mechanism that I described to you earlier. The conversion of what's called T4 to T3 simply involves pulling one of the iodines off of this molecule that has 4 and making a molecule over here that has 3. And notice I'm not giving you the names, so I'm not holding you responsible for those names. And the last thing there, melanin, the pigment, is made as a result of a polymerization of tyrosine. And you can see some of that polymerization here and points of polymerization where this can be a very, very, very large molecule. Depending upon how many of these are polymerized together, the actual color and darkness of the pigment changes. The more rings you have associated with each other, the greater the absorptive ability, and the darker the melanin will be. Melanin, of course, is the pigment associated with skin color. And so it comes from tyrosine as well. Okay, so a lot of cool things arising from tyrosine. The last, or family as such, histidine is also a family, but the last family I'll mention is the pyruvate family. The pyruvate family, of course, comes from pyruvate, and it leads to these 4 amino acids, and they're actually very easy. Alanine is a transamination product of pyruvate. I've referred to that earlier, so there's another transamination. The other 3 are what we call branch chain amino acids. So I'm not even going to ask you to memorize which ones are the branch chain amino acids, other than to know that the branch chain amino acids are also part of this family, right? So I'm not going to say is leucine a branch chain amino acid, or isoleucine, or valine, okay? But the members of this family include alanine and the branch chain amino acids. Transamination reaction, you can see here, there's your amine source again of glutamate. There's pyruvate, and this is the product afterwards. That's alanine on the right and alpha-ketoglutarate on the left. I'm going to spare you. I'll show you and hold you not responsible for what I'm going to show you here. There are the branch chain amino acids. And their synthesis actually is a little complicated. I'm not going to hold you responsible. I'll just let you admire the beauty of this figure. I actually made this figure myself, and it took me about an afternoon, so I'm happy that I've got to use it if I made it. That's the way I figure, so. Okay, well that gets me almost to the point of the end, but I'm going to, well actually no, I'll do one more thing. Then I'll do a song, and we'll finish the breakdown on next time. The last group is not a group, it's one molecule. It's one amino acid. And that one amino acid is made from an unusual source, ribose 5-phosphate. Ribose 5-phosphate, of course, is also used to make nucleotides. And so nucleotide synthesis and histidine synthesis overlap at the point of that molecule. After that point of that molecule, they do diverge, but there are 10 steps. It's a fairly complicated pathway, that is necessary to make this. The second enzyme of the pathway is feedback inhibited by the end product, which is histidine. So leave that there. Okay, let's turn our attention to the song for today. The song for today is about the end of the term. And one of the things you'll see about the song today is that it was written for another class back when I used to have it in the class in Milam Auditorium. So you'll see what that's about in a second. So let's go with that. Hall, it's 12:30 and Ahern's getting wordy. He walks to and fro while not talking slow, giving it to BB450. Was happy when the term got started. Lecture notes and videos galore. MP3s got added to my iPod. Recitations sometimes were a bore, and exams bit me roughly when the curve turned out ugly. I don't think it's so, my scores are too low, sliding by in BB450. An examination on December 9th, 6 p.m. I'll have my card packed with the information. I don't have to memorize it then, and I'll feel like a smarty, my jam-packed note cardy. One more to go, and then ho ho ho, I'll be done with BB450. See you guys for a fun Friday.